Thank you very much for your introduction. It's a pleasure to be here, and it's great to see so many people here today. And it's just like Bruce said, it's wonderful to be here. For finally, I can talk about what I'm most interested in the world without having to spend half a day explaining. So it's great. I've been constantly adding slides to this presentation for the last 3 days, and I come up with, oh, I have to say something about that. I have to say something about that. So we'll see how I will progress with. But at least I'm going to talk mostly about hyperspectral imaging. I started out doing spectroscopy, optical modeling with Lars. Starting from point spectroscopy, moving into hyperspectral, and that's where I kind of fell in love with the technique. And I'm trying to give you a little glance of what's going on, what has been going on in my group over the last years. So, but first of all, just to let you know, where am I coming from? So I'm from Trondheim, Norwegian University of Science and Technology, and some of you might wonder, why am I staying in the same university? It's because it's the best university. So, no reason to move. So, thanks to the map projection, and we're so far...
Universities in Oslo as well.
There is a university in Oslo, but they don't do biophotonics, huh? They do some. Yeah, I know.
Biophotonics is here from Oslo.
Yeah.
Yeah.
So, and thanks to the map projection, and we are so far north, and Norway actually looks big. So, it is a small country, it's far north, and we're not many people doing biophotonics in Norway, unfortunately, and that's why I'm so happy to be here, to be able to discuss with you. So, a little bit about the university, and what is great about this university is that this is a picture of our campus. I think Bruce actually been there. Mm-hmm. Yeah. My office is located just here with a beautiful view. And my view is down to the hospital, which is located here. So we're very closely integrated with the hospital. And we have a brand new university hospital. They actually finished constructing it last year. And they have one floor in every building is a university floor where we are doing research. So, and we have clinic underneath and below and over, and one floor for research in every building at the university. So it's some great possibilities. And for those of you interested in nanotechnology, we also have a great nanolab facility that opened some years ago. Anyhow, we're the second biggest university in Norway with 23,000 students, so it's pretty big to be Norwegian. Anyhow, that's not what I'm going to talk about. And just to, you were mentioning Lars, and I got this picture from Lars. It's actually a picture of the first ever biophotonics experiments done in Trondheim. And as you see, it's a patient here. It's a rat. And he was trying to coagulate brain aneurysms with argon laser. That's what was, and they did some heavy smoking in the lab to be able to see the laser in the picture. Anyhow, this shows that the history of biophotonics in Trondheim is actually quite long. This picture was taken the same year as I was born. So it has been going on for a while. And here's a picture of Lars. For those of you who don't know him, he's the greatest guy on earth, I'm sure. He looks like Santa Claus, and he's even nicer. However, he was a challenging supervisor. But if you made an effort, he always gave you the best possibilities ever to really expand your knowledge and work on interesting things. And that's the thing, and that's something I learned from Lars. Your research should be curiosity driven. It should be because you're curious about something, because you have a question, it's something you want to find out. And that has led us to working on a wide range of applications. So we haven't been only working on skin or only on psoriasis or port-Weinstein birthmarks. We have been doing everything. More or less, everything that passes by our lab is in the risk of being measured. And if you look at this list of topics, it's even something that does not fit that we started doing recently. It's actually this one. I just recently had some prayer books from the 15th century brought to my lab for doing hyperspectral scans. So that kind of even adds to the complexity of the field that we are working in. But here are some examples. Portwine stains, most of you probably know quite well. These are some of the first experiments that were done in Trondheim by Lise. Some of you might know her. Some cardio measurements. We actually gave that poor pig a heart attack. You can see it's a pig because you can see the leg here behind. Psoriasis, studying inflammatory reactions in skin. It's been a great interest. We also did some measurements on king crabs. So if some of you are interested, I have some really interesting information about the epidermis of king crabs. That's for more special occasions, I think. And of course, this one. I will get back to that one. And that's, I added it specially this just recently. It's about methemoglobin and methemoglobin formation. I think that might be of interest for you. I spent like half a year in the lab working with blood samples and heating them and boiling them and doing all kinds of nasty things to them. So, but just to get a little more picture of what are we actually supposed to be doing. Well, we want to try to discover new diagnostic and therapeutic uses of light, and of course also characterization. I've been doing a lot of characterization because I've been moving into all these strange fields like measuring bruised skin and things that no one else had been doing before. So then we had to do a lot of characterization because we had to kind of map up what's going on before we could start doing any diagnostics at all. But it's tissue light interaction, it's optical modeling, optical diagnostics, spectroscopy, and recently hyperspectral imaging. And of course, we want to end up with something that is useful in the clinic. And you probably know this is a hyperspectral data cube where you have 2 spatial dimensions, and in addition, you have a spectral dimension. So practically, you have a full spectrum in each pixel of an image. And that's kind of, I said, I fell in love with the technique. And what made me fall in love was actually this picture, or the processed version of this picture. This is a grayscale image at the wavelength where you have the best contrast between hemoglobin and tissue. It's taken from the outside of an aorta. It's not the inside, it's the outside of the aorta. And I was looking at it and, yeah, well, I can see the vasculature pretty well. Then I did the hyperspectral scans, and suddenly I ended up with this picture. And being able to see the vasculature this well, you could even see there are some tiny aneurysms here and there. You could see the vessel lumen, you could see everything, and you get almost the 3D effect. That was the moment where I fell in love with this technique. There are so many possibilities. If we can only manage to dig out the information, that's the tricky thing. We can measure so much, we can gather so much data that we could spend years and years and years analyzing it. So that's part of the trick, getting the fast, efficient analytic tools to bring us forward. So hyperspectral imaging. If you look at the imaging chain, basically this figure could be for any field. It could be food quality monitoring, it can be remote sensing, it can be anything. Basically you have an object, you shine light on it, you have some kind of hyperspectral sensor, which can be realized in a variety of ways. You get some raw data, you do some pre-processing and calibration until you get your data in your preferred format. Then you do your image analysis, you extract some information, and then you have a user evaluation before someone makes a decision to perform an action. So where in this picture do we work? Well, we're within the yellow frame. We try to do everything within the yellow frame. We try to develop the cameras. We try to make the pre-processing algorithms, the image analysis, and also to extract the useful information. So you could say it's pretty ambitious, but I think it's good because then you know the complete, you're not working on only one small part. You're actually trying to follow the whole imaging chain. And if you look at what are the different components here, the sensors, we don't make the sensors, we just use them. We buy them off the shelf. Sometimes we have trouble buying them because they're often military technology that is not allowed to import into Norway. But anyhow, we work quite closely with industry, developing camera technology, especially fitted for optical imaging of biological samples, like in low light imaging conditions. And of course, making the setups, doing the characterization, the modeling, the image analysis, and of course, hopefully, we will end up with clinical systems that are actually useful. And we're about to get there. I've been working with hyperspectral since about 2005. It's almost 10 years, and now we're getting close to our first clinical system. We are testing the prototype, the first real prototype, now in November. So this has been a long path. And just to show you, I said that hyperspectral imaging can be done in many ways. You could use filter-based systems. You could use mirror systems. You could use Fourier transforms. You could do whatever. It's many ways to realize a hyperspectral system. The idea is that you have high spectral, high spatial resolution in the same system. In our system, we have adapted cameras from remote sensing. In remote sensing, they are usually flying with the cameras. They drill a hole in the bottom of a plane, mount the camera, and they fly. So when we are using these cameras, we have to do scanning. So we have to do some mechanical scanning. We have a two-dimensional sensor where we have one dimension for the spectral information and one dimension for one spatial coordinate. Then we have to scan the camera to get the second spatial coordinate. And this is just a short figure to show. First of all, you need the scene. This is my former PhD student who made the figure. So first, you have the incoming light. You have a close-up lens. Then you use focusing mirrors, elliptical mirrors, to focus onto a slit to select one line of your scene. And then you have another collimating mirror. You have a dispersion grating, some lens optics, and then the detector. So yes, you disperse the signal, and then you project it onto the sensor to get a map showing the spectral and the spatial, and then you have to scan. So I'll skip this one. So just to show you the specifications of the camera systems that we have today, we are covering a wavelength range from 400 nanometers up to 2.4 micron with two different devices. This is the sleeve camera. Oops. This is the sleeve camera. It has a spectral resolution of 3.7 nanometers over the wavelength range 400 to 1,000 nanometers. It has quite good optical quality. It has been focused on minimizing optical aberrations. It has a low smile and keystone effect. You know that you're actually measuring the point where you're supposed to measure. And in addition, we have several lens systems so that we can image with, you see the resolution here, pixel size. We also have another lens that is custom made that gives us about 30 micron spatial resolution. Yes. So hyperspectral, what is it all about? It's about digging out information that we cannot see otherwise. So this is a little task just to keep you awake. I'm wondering, which plant is artificial? Any ideas? It's one fake one. Anyone want to guess?
The third one from the
third one from the left. Any other suggestions? Huh?
Obviously not right.
Okay,
I'll stick with the first one on the left.
And there is a missing person in the jungle. Can you see the missing person? But there's some really some experienced people here used to doing visual inspection. Usually people don't have a clue. If we do some image processing, David got it right. This is just by looking at the spectral signatures, you could easily single out that plant. You cannot see it with the bare eyes. And the same thing with the missing person. By doing some anomaly detection on the spectra, you can easily find the missing person in the jungle. But it's not, and this is what it's all about. It's about helping us to do better. We want to see what we cannot see. That's what we are doing. We are trying to enhance features that are not easy to see by visual inspection. And of course, it works because you're looking at the spectral contribution, and you can see that this Lego man has a spectrum that is significantly different from the background. So you just run a basic anomaly detection based on the spectrum, and you'll find it. And of course, you could also use this for medical applications. It's not only for finding fake plants and Lego man. It's also about finding features that we are interested in. So, what's our challenge? Well, you know it quite well. It's about the mixed signals from many observers, having a big mixture of everything. The scattering materials that give us trouble. And one thing that I've been wondering quite a lot about is the two lower ones. That the depth and surface signals are different. You cannot really look for the pure chromophores. It will never work. You will never, with image processing and classification based on known spectra, it will never work. It will work on the surface, but it won't work in the depth. It's like, we know blood is red, but our veins look blue. It's all about tissue optics and the error introduced by scattering. And also that the penetration depth varies with wavelength. It's something that might be a problem if you do your fitting all over the wavelength range and you compare apples to bananas, because you have such a different penetration depth. But it might also be a resource, and maybe you can use it to get some depth information, because that's what we have been trying to do. And of course, I had to bring my favorite spectra. You probably know them quite well. I have the oxy and the deoxy, and my small little friends, the deoxy dromedarian and the oxy camel. I use them when I teach this for the students, and they always think, oh, I remember the camel. So what was really going to emphasize was this black line here, which is a close friend of mine. It's called methemoglobin. And actually, the first time I discovered methemoglobin was a mistake. Someone has been putting my blood sample into the freezer, and that did not turn out well. So when I was measuring the blood sample, something very strange occurred. I had a peak at 635 that I really couldn't make sense of. And when I started looking around, I discovered methemoglobin. And methemoglobin is formed as a result of oxidative stress. It might be, as in my case, freezing. It's also teared up the cell membranes and stuff. It might be heat. That's why I've been interested in it, in connection with laser therapy and laser treatment, where it's actually formed. You can see it formed during PDT. And when you use ALA and you use 635 as your excitation wavelength for the PDT treatment, it might actually mess up your dosimetry because it reduces your penetration depth. And if you look for longer wavelengths, the absorption for longer wavelengths than I have plotted here, the absorption of methemoglobin is about 4 times the absorption of hemoglobin. So it's also something that can be utilized if you can just control the formation of it and you can detect it precisely. And if you're working with burns, you will certainly have methemoglobin in the crusts. That's where I actually found the best data available was from burn fire victims, where they have extracted blood samples and looked at them. They had almost completely methemoglobin formation. But as I said, it might also form during laser therapy. And the interesting thing is that EMLA, the topical anesthetic, is also contributing to formation of methemoglobin. So if you combine those two, you might have a really increased methemoglobin formation. And you can survive with up to 30% methemoglobin in your blood, but not all people, because there is a genetic deficiency in an enzyme that converts methemoglobin back to hemoglobin. And some people have this deficiency, and for those people, that's not a good combination. I've seen one case report, I think it was from Florida, where a woman actually died after hair removal because they have treated large areas on her thighs and they had used massive amounts of EMLA. And she had this genetic deficiency and she didn't know about it. So the poor woman died. So it's something to look out for. I've seen it once. I've seen increased methemoglobin concentration once in a person where I measured the skin. I was measuring bruised skin, and suddenly I saw this funny signal in the normal skin, and this person did not know. But it was confirmed later that it was actually this genetic deficiency. So then you need to take some precaution in what you're doing. But that was a little sidetrack, but anyhow. And just to show you how the absorption changes, these are some really, I think these measurements are 15 years old. It was something I did this summer. I stayed in the lab. It actually shows what happens when you heat hemoglobin in solution, in water solution. And you can see here, there is a peak showing up at 508. There is another peak showing up at 635. And the oxyhemoglobin peaks here will disappear slowly. And the maximum formation of methemoglobin will occur. There is a trade-off between methemoglobin formation and coagulation. So according to my measurements, the maximum formation of methemoglobin, the maximum formation rate is at about 72 degrees centigrades. So, and you also see, if you look at the shorter wavelengths, you also see a shift of the Sorey band peak. So, well, this is what we are hunting for, usually, the skin spectral signatures. And many people think it's like black magic when you start interpreting these spectra. And to a certain degree, it might be. What I wanted to show you here was the effect of pigmentation. So this is me, and this is Eivind, my former PhD student. And you can see, well, these two spectra are measured on my skin, and this spectrum is from Eivind's skin. And the difference is pigmentation. So it's quite a strong effect. So that's a reason for moving to longer wavelengths, which I'm going to talk a little more about later. It's difficult if you're interested in things like bilirubin, then it's difficult to move to longer wavelengths, so then you have to find actually other chromophores to work with. Another thing that I want to point out here, these two measurements that were measured on me, I have a tendency to perform experiments on myself. So the difference between those two was that my office is at the 4th floor, my lab is at the basement. I was running up twice, up and down, before doing the second measurement. So you can see the oxyhemoglobin has, I'm blushing, it's measured on my face. But this one, I also want to point out this one. And if you think about your chromophores, the first thing that occurs to you is probably bilirubin. But I don't have jaundice. So this is actually beta-carotene. And the reason why I know it's beta-carotene is because it was an intended experiment. I read a paper that it should be possible to measure beta-carotene in skin. I measured them myself and I couldn't really see it. So then I started my carrot treatment. I was eating about 1 kilo of carrots every day for 3 months. I got terribly bored of carrots, but obviously very healthy. And then the beta-carotene peak turned up. The thing is that beta-carotene, you find it in the stratum corneum and you find it in the subcutaneous fat. Well, I'm not dieting, so my subcutaneous fat stay where it is. And the signature is still there. Many years later, I'm not getting rid of it. So, and if you see me in a little blue light, I seem a little bit orange. So, and that's exactly what happens when you take these tanning tablets with beta-carotene. I think one of these tablets contain enough beta-carotene to match up like 17 kilos of carrots. It's like a big bag. Anyhow, I think it's a nice example. And then I'm going to say a little bit about modeling, because we're not only looking at these spectra, we're trying to derive some information from it. And when we were working with reflection spectroscopy with Lars, and also here, it was developed a lot of algorithms and models for diffusion theory modeling of reflectance spectra. But this gives us an additional challenge when we move to imaging and we move to hyperspectral imaging, because are these models directly portable to this new world of imaging? No, they're not. We have to make some assumptions about the pixel size. How is the pixel size affected by the penetration depths and so on? So there might actually be an optimal pixel size for imaging based on these diffusion theory calculations. But anyhow, we have an inverse model. Based on that, we do forward modeling. We compare that to the measured spectrum iteratively using a least square fitting. And we fit quite a number of parameters. But we, as I said, we try to stay, SI means semi-infinite. We try to stay within 2 main wavelength regions. And the reason why is because of the difference in penetration depth. So here we are fitting oxyhemoglobin, deoxyhemoglobin, methemoglobin on carboxyhemoglobin in case we have any smokers. And we tend to have smokers, and they might have up to 15% carboxyhemoglobin in their blood, which is pretty nasty. They tend to look pretty healthy because it gives the skin a nice pink hue. So they look healthier, but they're not. And you could actually see it in people that have committed suicide by inhaling exhaust. They die from carboxyhemoglobin or carboxy monoxide poisoning, and then they actually have pink spots all over. And we are also, up here, we also fit hemoglobin, oxyhemoglobin, water, and also, of course, lipids that are not in this list. And we do this for hyperspectral images. And in our camera, we have 1,600 spatial pixels, and they are collected line by line, as we have a line scanning system. And one thing about these optical systems, to be relevant in the clinic, they need to be fast. It doesn't matter if we have the best results or the best data or the best models if they're not fast. The clinicians want a result like that. They have 10 minutes to see their patient and to make a decision. They will use other modalities if we cannot deliver within that time. In our system, we have an integration time, or not only the integration time, but typically 30 milliseconds per line for gathering the data. So the collection time is 30 milliseconds per line, 10 of it being the integration time. 10 milliseconds integration time is typical. And then, okay, our real-time limit is 30 milliseconds. We have to do all the processing we want to do within those 30 milliseconds. That means we have to do the inverse modeling. we have to do pre-processing, conversion to reflectance, we have to do noise removal, and all other kinds of processing that we want to do within 30 milliseconds. So how do we deal with that? We do it by using parallel processing and GPUs. And I think this model is very well suited for parallelization because it's the same thing that you should do for many wavelengths, that you should do for many pixels. It's perfect for parallelization. And actually, the inverse problem for 1600 pixels, 160 wavelengths per pixel, we can do it in 8 milliseconds. Of course, we have to cut some corners when it comes to accuracy. But what we are interested in is mainly relative changes and relative differences. So, and this is showing the quality of the fit. Of course, you can have as good fit as you want if you have enough parameters. With more than 5 parameters, you could fit an elephant. But we are sure we are trying to make sure that the parameters are within the physiologically relevant limits when we do the fitting. And we put quite strict constraints on which parameters are allowed in the fitting. And this is the timing. Just so, oh, it's actually 3.5 milliseconds now. This is the last figure I got from the student working on this. So we still have a lot of time to do all the other processing within. And the difficult thing actually turned out to be estimating the melanin, as we have such a strong crosstalk with the deoxyhemoglobin. So here's an example of that model. This is an image showing the melanin absorption coefficient at 1 wavelength. And here is an image. This is a healed ulcer. So you see the ulcer is here, and you have a very nice hyperpigmentation. Or maybe someone won't say it's nice, but it's nice because it gives us some contrast. You have some scar tissue here. In the middle, you have some blood vessels, some superficial vessels. You also have some deep vessels in the background here. So this is the melanin map. You can see it's looks fairly reasonable. You can see the hair standing out as darker, and you have this patchy appearance that you also see from the RGB image. And here is another. We also estimate the blood volume fraction in the dermal layer. And here is, we do it for different wavelength ranges, which gives us also some kind of depth information. You can say something if it's, at least we know that this light does not penetrate that deep. And here we can see the superficial vessels standing out with a higher blood content. And for the deeper layers, we see the blood vessels. Of course, this looks fuzzy because of the scattering. And also an example of the oxygen saturation monitored at 2 different wavelength intervals. And that's where it starts to become interesting, because that's very useful. And I remember the first time I tried to publish data on the oxygenation in this wavelength range, and at first it was refused because my values were too low. They were around between 50 and 65%. They said, no, oxygenation in skin is 80%, period. But it's not, right? And it's kind of logical that it's lower in the more superficial layers as you have more extraction of oxygen there. It has actually been consumed more. And actually, it turns out that this is a very valuable parameter to monitor. And we have a collaboration with Ullevål Hospital in Oslo, where they are monitoring critically ill patients with the risk of having sepsis, people that receive ECMO treatment. And by monitoring the superficial, the oxygenation in the superficial layer, the microcirculation, they can actually predict very early which patients are going to be septic. And they see the tiny little microhemorrhages forming, and they also see changes in the oxygen saturation. And it seems like this is actually something that happens quite often because the microcirculation is the first thing to shut down when something's wrong. So it's a very early prognostic factor. So, and this is also an example. I think it might be my hand actually. Oxygenation and blood volume fraction. This is based on the reflectance measurements of hyperspectral reflectance. Yes. And we've also been struggling with all these pre-processing. We had a problem that the images were not at focus. Okay, what do you do then? Then we develop an autofocus system that makes sure that we keep it in focus all the time by measuring the distance to the surface and profiling the surface before we get there. And then we had the problem of noise. Because if you want to do spectral unmixing, if you want to do image processing, if you want to do classification, you want noise-free spectrum. Everyone wants to get rid of the noise. So we start out with something looking like that, pretty noisy. And we have been using the maximum noise fraction transform to remove noise. It's like PCA, but it's a two-step PCA where you normalize or whiten out the noise before doing the second step. And it's actually turned out to be a very powerful tool. The problem is it involves a lot of matrix manipulations, which means it takes time. And it's computationally demanding. And you have to have the whole image before you can do the transform, because it will change depending on what's actually in the image. So we developed instead a line-by-line MNF transform so that we can do it as we are scanning. And what I'm trying to show here is that the results from the line-by-line and the full picture MNF are the same.
Is it a fairly linear operator? Is there anything to worry about, like the level of reflectance being modified because of a nonlinear feature?
Yeah, so we have tested this, and there is a bigger error in the beginning, but it evens out quite fast. So when you get, because we kind of update, what is the problem is to estimate the noise, and to calculate the covariance matrix of the image. And when you come up to like 50 lines, you're good. So here's just to say something about the timing. This is typically a conventional MNF, and this is a technology that has been used in remote sensing for a long time. So it's very well-known algorithm. And to start with, it takes like a minute to, say it takes a minute. You want to have the full image, you want to do the pre-processing, and then you have to estimate the noise and do your image statistics and solve a big eigenvalue problem, which involves a lot of matrix transformations. And it takes an additional. So you have a time lag of about 1.5 minutes. With the line-by-line way of doing it, you could do it within 8 milliseconds. So that means we're still within our 30 milliseconds deadline. And it works. That's the best thing. So here is the effect of it. Here is just to show that the line-by-line here is actually answering the question you had. Here you see 50 lines, 100 lines, 150, and so on. And as you see, the MNF and the line-by-line, they are the same. And why did we end up doing this MNF transform noise removal? It was because we tried all methods. We tried wavelets, we tried other transforms, we tried low-pass filtering, we tried averaging out, but you tend to lose something. You tend to lose spectral resolution or spatial resolution, and we didn't want to compromise those. We wanted to keep them. And this is an example. Here are some fluorescence data before noise removal and after noise removal. And it's a lot easier to work with these spectra here. And actually, we have tested it, what happens at the edge. If you have like very strong contrast changes, it seems to be pretty robust also at the boundaries. So, and I'm not going to talk a lot about, now I'm going more into the applications. I'm not going to talk a lot about bruises, but I have to mention it as it was kind of my baby. I think I was the first one to start doing spectroscopic measurements of bruises and start doing modeling. And we made a model to predict the reflectance spectrum from bruised skin as a function of time. And the thing is, we have been working a lot in the visual part, but in the later years, I found out that the severe part of the spectrum is at least as interesting when it comes to bruises. Because there's so much going on. There is so much edema, there is so much fluid flowing around that you could actually not detect in the visual part. So these are just some images to give you an idea. I love paintball. I really love paintball. I don't love the pain, but I love the bruises. And this is actually a paintball bruise from my arm. And my skin is very nice because it's so pale and non-pigmented and easy to handle. But you get these very nice donut-shaped bruises. And the reason why, we've done the biomechanics as well. So the reason why is that underneath the paintball, when it hits, you get compression. The tissue is compressed. But at the edges, you get shear forces. And the shear forces tear the vessels. So that's where you have the hemorrhage. For those of you interested, I have a beautiful video showing the waves when the paintball hit and the oscillations. It's perfect vessel functions. Really beautiful. Just to show you a little bit what I'm talking about, I'm not going to go into detail about this, but the nice thing is that if you compare different skin types in this wavelength region, the picture might suddenly be completely opposite. As you see here, the reflectance from a skin type 4 is actually higher than the reflectance from a skin type 2. So the pigmentation is not a big issue here. And here you see the water peaks. The water peaks maps very nicely to the reflectance spectrum. And someone asked me earlier today if it was worth expanding, going up to longer wavelengths to monitor water, and or if you could do with the 970 peak. And my answer is yes, I think so. And the reason is a bit complicated, but I do think so. And the reason is because if you start looking into the shape of the water peaks, that's also important. And the reason why that is important is because the shape changes if the water is free or bound. And that affects the different peaks differently. So you need to have more than one peak to be able to actually say something about that. Something that we were also discussing earlier today is that you might use the water peaks also to estimate temperature by looking at the vibrations and the shape of the peak. Of course, that will involve a lot of modeling, and that's my little pain at the moment. There are no good collections of optical data in this wavelength range. I'm missing the optical properties to do proper modeling. And they are scattered, only scattered data are available, and they are not very good quality. So I wonder if we have to do the job ourselves to get the data we need for modeling. So here are some more data from a student being shot, having bruises, and here's how we scan the arm, some vessel extraction, and of course this is some very simple image analysis, but you see the bruised area here. And this image it's actually one of my favorites. Because here you see the bruise, you see the small zone underneath the paintball where you had the compression, and you see this red area around. The interesting thing here is that this is immediately after firing the paintball. So what you see here, the pink color, this is of course not the real colors, but the pink color here, you see the same thing in the blood vessels. That's blood accumulating in the area. What happens here is actually the edema that is forming, the tissue fluid that increases the pressure in the area to stop the hemorrhage. So you could actually see the difference between the tissue fluid and the blood, which is pretty interesting. And that's in the Swede range of the spectrum. Yes, and also just to show you a couple of measurements that we've been doing. Some fluorescence measurements. We measured on something that I call my cheesecake collection, where we had phantoms made. They are not made from intralipid. They're made from corn oil, actually. I had some biotechnology people to make them, and the optical properties actually match the skin very well. You have to have these micro bubbles to get the right scattering. But anyway, they work fine. And this is just adding a fluorophore to see that we are able to detect what is happening. You can see the absorption. We can see the fluorescence emission from Alexa here, in this case. But then to the real world. And we did some fluorescence measurements on some aorta samples. And this is actually, it's not a pizza. It's an aorta. It's how your aorta will look if you eat too much pizza. So here, what we can see here, these are all autopsy samples. So this is persons that actually died from some cardiac event. So they were already having some problems. What you can see here, the yellow part are lipid deposits. This is actually a ruptured plaque. So it's kind of a wound. You have another one here. These are vessel exits from the aorta. The interesting thing was when we did fluorescence imaging, hyperspectral fluorescence imaging of this sample. It looked like this. And I think this image is so beautiful, so I include it in all my presentations. and this is actually raw data. So you could see it's almost no noise because we kept tuning the instrument for about a week until we got there. But anyway, you could see here you have beta-carotene, here you have collagen, here you have some blood masking the fluorescence. And if you combine this with the visible image that we also collected, you can do classification and you could look at single plaques. And what was the idea here was to say something about the heterogeneity of these plaques. Because if you biopsy them, you usually take a biopsy in the middle. And then you say something, okay, this is stable, this is unstable. But it turns out the picture is more complicated. Because this plaque here, this orange region here, is stable. It has a nice, thick, calcified cap, no problem. But this purple region here and the green region, they are definitely unstable. Especially the purple zones here have a very thin cap. It might rupture any day. And that's exactly what happened to that patient. So it's just to get a broader picture of what is going on. The spatial resolution in this case is extremely important. And we also did biopsies here. I think we had like 6,000 slices from one plaque. So we really know that this is the right pattern. Also thought I would show some images of small animal measurements with the hyperspectral cameras. And this is a study we did to look at the toxicity of dental materials and also the effect of the light from a dental material curing lamp compared with the UV light source. And we used mice, angry little creatures, black mice about this size, and they were biting and jumping and they didn't want to get their teeth treated. Anyhow, it's possible. Even here, you see this little mouse here is receiving gas anesthesia with a mask. And we are actually treating the mouth of the mouse here. So we were actually working with the tongue and the lips of the mouse, and we were comparing with skin. So it's possible, even though the instrument is rather bulky, it's actually possible to do measurements on live animals, and with a fairly good resolution. And these images here, they are RGBs constructed from the hyperspectral images, just to give you an idea about resolution and so on. So here is immediately before, this is immediately after light exposure. This is just light, blue curing light, with a fairly high dose. And this is 1 day after. 24 hours later, the tongue collapsed. And of course, we had to sacrifice this animal because it was not good for the animal. And anyhow, now we have the images. We have the possibilities to go in to which pixel we want and pick out the reflectance spectrum. And in this case, these are the average spectra from a region of interest selected at the tongue here. And you can see what is going on. Well, I suspect here, see, I've done the calculations, so I know the answer. But you see some methemoglobin formation, you see some deoxygenation, you see some scattering changes, you see some changes in water content. So there is a lot going on. And we are not done with analyzing these data yet. So that's why I don't have any numbers, just to show you the possibility. And then I will talk a little bit about one of the European projects that we are involved in. It's a project with 6 partners in France, Germany, Estonia, and Norway. And we are trying to develop optical diagnostic technology to diagnose arthritis in the finger joints. And we are looking especially at the early stage arthritis. And the reason why this is critical is because joint destruction starts early. It starts within a few months. And it's difficult to see the difference between rheumatoid arthritis and psoriatic arthritis. And what the clinicians do is usually to look at the family history, to look at the patient's history, and then they make a guess. And then they start treating the patient. And they tell them, oh, if you're still painful, after like 3 months, you should come back and we should switch your medication. But then it can be too late. And so far, the current methods of diagnosis is usually they do ultrasound to look at the joint capsule, to look for angiogenesis and new vessels. They measure the blood flow using ultrasound. You need to be trained to do that. And all the physicians get different results. So you can only compare within one physician. If there is a different person looking at the same patient, you will get a different result. And you can also use MRI. MRI is a good technique, but it cannot distinguish between rheumatoid arthritis and psoriatic arthritis. The same with X-ray. You can see joint destruction, you can see what's happening, you can see the consequences, you can't see the reason. So basically, there is no method. So we thought maybe we should try using optical technology. And we are trying to combine hyperspectral imaging with photoacoustic imaging. So the Germans do the photoacoustic part, and we do the hyperspectral part in this project. And what I'm going to show you now is some actually patient results. We did a measurement on 15 patients with known established arthritic disease, both psoriatic and rheumatoid arthritis. We also had a control group of 20 people in addition with no disease. We did all the standard evaluation, the clinical evaluation, the ultrasound, and we also had MRI and X-ray. And we looked at the MCP and the PIP joints, which are using these joints. And well, we started out doing reflectance. We thought, we will do reflectance. But it turned out to be a problem because we only saw the superficial things going on in the skin. And that's not where the disease activity is going on. The activity is going on inside the joint. So then we decided we have to try transmission. And actually, it turned out to work quite well. So here, I'll just pop up all of them. Here, you can see we could actually see the joint here. You can see the joint capsule. This joint, this joint, this joint. In the control, all the joints are clearly visible. It's a little more difficult in the patient though. But we are able to see nice transmission curves. The funny thing in the patients is that the peaks, the transmission peaks are shifted. They're not exactly at the joint. They're shifted somehow. We are really wondering why, because we see it systematically. That the peaks do not appear exactly at the joint. They are shifted. And, but there are some differences in transmission. It gets a little more fun. Oh, this is something else. Okay. I put some slides in between. I will get back to the transmission measurement. The thing we did to handle these data, because we were not familiar with hyperspectral transmission data. We didn't know what to do with it. We needed to extract some features. We had lots of data. We started out doing some Monte Carlo modeling to model the transmission through the joint to see what we can expect. And a 3D Monte Carlo model. That turned out well. It's in accordance with the experimental data that we get. But still, we want features. We want to see what is going on in these arthritic joints. What we know is that they are hypoxic inside the joint. That's why they're forming new blood vessels. But we also know that they have increased blood flow outside the joint. We know that something is happening to the synovial membrane inside the joint. It might thicken. We also know that the synovial fluid inside the joint is changing. Because you get cells in there when you have active inflammation. When your joint hurts, it can be an infection, it can be an injury, it can be rheumatoid arthritis, it can be psoriatic arthritis, it can probably be a lot of other things as well. But at least we have at least those four. And it turns out that the cell count in the synovial fluid is a quite good prognostic factor. And then you can say, why can't you just take a sample? Because the volume of fluid inside a finger joint is so small that you will never ever be able to get a sample out. But you could measure it with transmission. You could measure the changes in scattering associated with the difference in cell count. And in addition, you could monitor the oxygenation around the joint, in the joint. You could look at the diameter of the blood vessels to see if they are changed. And actually, what they do, there is a technique where they are injecting some fluorophore, and they are looking at the blood vessels. And then they are actually looking at the blood vessels in this region to see if they are increased. And if they are, then it's some diagnostic value in that factor. But we wanted something more specific. So we started looking into other tools. What other tools do we have in our toolbox? And one of the tools we found was wavelets and wavelet analysis, which turns out to be pretty good for extracting information about vasculature. And just to show you a little bit, wavelets is actually in the spectral domain, is where you actually are sliding a known spectral shape across your spectrum, selecting small parts at a time. So it's a filtering, it's a kind of combined high-pass, low-pass filtering at the same time. And we did some occlusion measurements. This is again my arm. In case you want to know, there is the mole that you can see. It's always good to bring the sample along in case someone wants to do control measurements. And here are the spectra from some selected regions. Oh, this is my student putting together the figures. So this is the kind of normal skin. This is the mole. This is the mole. And this is over this vessel here. So you can see the spectral changes that you expect to see in skin. You could also see my beta-carotene is still here. And you can see the deoxyhemoglobin, you can see the oxyhemoglobin here, and the deoxyhemoglobin during occlusion. But some other thing that is quite interesting in my skin that I actually haven't seen in any other is that here you can see the water peak, 970. You could also clearly see the lipid peak at 930. I haven't seen that in anyone else. Maybe because I'm the only female in the group. That men have less subcutaneous fat. But at least it's pretty visible. So how does this look after we run it through the wavelet algorithm? So these are some examples of what you can get out. Here are some images showing what I believe are the deeper vessels. And here, this is another interesting feature that you don't clearly see, in, at least not in the RGB images. And I wonder if these are arteries. I'm not quite sure, but I think it might be. And you can see they are disappearing during occlusion. Anyhow, the wavelets turn to be a very good way to extract information about the vasculature. And another funny thing was that we found that two of these different wavelet components, if we subtracted them, we got a very nice melanin picture. That was just a coincidence. We were just playing around with the data and then look pretty similar. It's just the mole that is different. So it seems like it's also a very good way to extract information about melanin. And we haven't really tested this carefully yet, but we will. And if it's a good way to estimate melanin, I will be very happy because I'm so tired of the crosstalk. And how do these wavelets then work on these arthritic patients? This is a typical patient with arthritis. You can see there is swelling. And you can see some dark areas here. That's actually inflammation picked out by the wavelet algorithm. And this is also wavelet extraction of, in this case, blood volume. And here you can see this is the inflamed joint. You can see there is a lot of blood accumulating around the joint. And this is the other hand of the same person. And this is the region where they look for increased blood flow using this fluorophore. But in this case, there are no additional chromophores added at all. It's just the blood itself. And these are some transmission data. This is a patient with psoriatic arthritis. And you can see the transmission here. You can see the joints. You can see the blood vessels. And the fingers are quite easy because we know where the blood vessels are and we know what they're supposed to look like. So we know that we have the right number of blood vessels and that we're not only seeing the superficial ones. But we do get quite good pictures. And in this case, we have not used any enhancement algorithms or filters to extract further the vessel structures. Of course, you can also do that. This is a patient with rheumatoid arthritis. As you see, there is something really severe going on in this joint. You could also see vessel structures here that are stretched on the sides here. And this is a normal control. You can see you cannot really see much in these patients. So the thing is, when these are just transmission data, when we apply the wavelets to these patients, we can clearly pick out those who have rheumatoid arthritis and those who have psoriatic arthritis based on combining different wavelets, which is pretty nice. I was really happy when I got those data on my desk. So that's about it. I'm going to show you a little video. In the end, just to thank all the people and all the funders, all my students, and of course Lars. And this is what it looks like in Trondheim now. We have been having beautiful northern nights the last weeks. Yes. So that's the benefit from living at 64 degrees north. So I'm just going to show you a small little video. It's actually a part of a video, and there is no sound because it's in Norwegian. but it's just to show you how the scanning system that we have used to gather the hyperspectral data from the arthritic patients, how it looks. This is actually a video that tells something about how it is to be a professor. So, so this is the scanning system. We have the light source for transmission underneath. We have 2 cameras mounted above. We have a Lexam plate where the patient is resting their hand. This is me positioning the patient. And you can also see the scanning speed here. So it's pretty fast. And all the processing is done while scanning. It's just this is some very preliminary. I had to make some pictures to that looked pretty for them to show. I'm really excited about this. You can tell, right? So yeah, I think this is. I'm telling the story about arthritis. I think this is it. So thank you very much for your attention.